everybody to come here this afternoon. As you have been told, I'm going to be talking about Expression Atlas, which is a platform for integrating and displaying gene expression data. So first things first, what is Expression Atlas? So it's a collection of web services and online resources that provides gene expression patterns associated or in response to experimental conditions. Such experimental conditions might be a phenotype, a specific phenotype, or a disease, a treatment, a response to a compound, etc. So these are some of the things that we offer. You can find expression of a gene or a set of genes that share a common feature. You can search for phenotypes using experimental factor ontology expansion. There's also a search for gene set enrichment analysis that uses an API and provides a graphic display of the results. And also you can find co-expression patterns in an experiment. There are R objects that you can download and integrate to your R session. We offer also pre-computed clustering analysis in PDFs and many other things, of which I'm going to show you a few highlights first. So to give you an idea of and some numbers, we have over 3,000 experiments. One are like the usual suspects such as GTEx, the Human Protein Atlas, ISCB, ENCODE, and others. Unfortunately, it uses a font. So I'm going to switch to the PDF, if you don't mind, because sure. Sorry about that. Okay.
Control L?
Yeah.
Okay.
So is there a way? Okay, I think that's not very happy with the interface here. So, yeah, just to show you, I guess this is the only slide where I'm using the font. So this is across more than 40 species, not only human and model organisms such as Arabidopsis or C. elegans. We offer also experiments about other mammals, birds, insects, fish, plants, yeasts, fungi. So let me switch back here. And this is a more detailed breakdown of the number of experiments that we have in Expression Atlas. Of course, the species which gathers more interest and more requests is Homo sapiens and mouse and the usual, I guess, the more common model organisms. Now, of course, everything starts with data. So where does this data come from? So researchers worldwide, they produce their studies with RNA-seq or microarray experiments. They use Annotator, which is a tool developed by some members of our team. And they use that tool to submit data. Then we have our curators that verify that the experiments meet several quality requirements, and then that's put into ArrayExpress, which also integrates studies from proteomics experiments and also from Geo. Now, this is a great simplification, as Laura probably can tell you, and she can, of course, if you're interested, she would tell you all the ordeals that she and the rest of the curators must go through so that experiments are properly curated in Expression Atlas. So some of the things that we do before every release is to get the latest reference genomes from ensemble and ensemble genomes. We also reannotate the experiments with the latest version of experimental factor ontology, the Uberon as well. Also verify that the anatomical systems for which we provide filters are in a good state. And this is a semi-automatic process. So there's this tool called Zuma that our curators train, and with every release it gets better, but still it needs quite a lot of human input. There are two big groups of expression data. One is baseline expression, which is bulk RNA-seq or proteomics experiments where expression is measured in absolute terms, either in TPMs or FPKMs. And there is differential expression, where the expression is provided as a log-fold change of a contrast between one biological condition and another. Finally, like most of our experiments are differential expression, but in terms of raw data, baseline experiments are the most challenging ones for visualization, storing, and processing. Now, this is the URL that you, if you're interested in going to Expression Atlas, and this is the web page that will greet you. And as you can see, we have a gene or gene property search. This is where you will input either a gene name or a gene ID or a gene features such as zinc finger or an ontology accession such as an interpro or also a reactome term, and that will give you back the results for the genes that match that feature. You can also narrow by species. So let's say there's a gene name that matches several species. There you can actually say, okay, I want this gene name, but only in mouse or human. And here you have the ontology EFO search input, where you can search for any terms of the experimental factor ontology. Now, I want now to give you a bit of a break and talk to you about something that is very fitting to Prague, in this beautiful city that we're in. And it's about Franz Kafka. So Franz Kafka was raised and born in Prague, and unfortunately he died at the very, very early age of 40 years old, and he died of tuberculosis. So as a case study of things that we can find in Expression Atlas, I thought very fitting to search for studies related to tuberculosis. So this is what you would get if you start typing tuberculosis in biological conditions. You can see that's the experimental factor ontology tree, and you can have also different types of tuberculosis, but for this case, we're going to just narrow on just tuberculosis. And this is the results page that we arrive at. So as you can see, on the left you have facets and filters where you can actually narrow down your search by species or by experiment type or number of replicates. And also you can download the results. And at the bottom there's a human result. So if we click on the gene name, which is PTGS2, we would jump to the gene page, which has three main tabs: baseline expression experiments, differential expression experiments, and an information about the gene. So this is the baseline expression tab, and again on the left you always have filters that you can use to show and hide the information that you're interested in. And on top of every heat map, there's also some filters and sorting options for narrow down on the genes and factors that you're interested in also. If you click on the information tab, this is some information about the gene that you just went to. And if you go back to the baseline expression, you can also click on one of the experiments that have been found to match the gene. and that will take you to a base, to that experiment already filtered by the gene that you search for. This is accompanied by an anatomogram illustration that you can see that helps you correlate the tissue to the place in the heat map where this tissue, where the gene is expressed and in which tissues. And also, we offer another filtering option in which you can choose specifically which columns, in this case tissues, you want to show. You can go about individual columns, which is the top block, but you can also go for more coarse-grained options in the tissue supergroups, and that will check or uncheck groups of tissues. Now, there's also another alternative visualization, which is a box plot, which is, here's an example of that. So this is like a very quick overview of some of the things that Expression Atlas provides. But of course, this is a technology track, so right now I want to switch to technology a bit and show you what happens under the hood whenever you perform one of the searches. So, of course, it all starts with a browser and URL request, and that URL request is received by a Tomcat server. The Tomcat server matches the URL that you have typed to one of the many code paths available, and then it queries 3 different data sources. So we have a PostgreSQL database, we have files, a storage of files, and then we have a Solr server. So in this case, everything except the database is managed directly by the Atlas team, and the database is managed by the EBI database team. Now, what are the 3 sources of data for? So Solr helps us in producing autocomplete suggestions, also in giving you the gene card information that I showed before, and to search for expressions using either metadata of different kinds, such as we saw, a gene term or, for example, disease, etc. The Postgres database is used to provide experiment metadata. So when you go to experiment page and you have a header of the species, the title of the experiment, PubMed IDs associated to that experiment, that's where that comes from. And also it has some other metadata such as design elements and also a list of expressed genes in each experiment, which helps when querying within an experiment page for a certain gene ID or gene name. And finally, the file storage is used to provide the raw expression data, so the expression values or the heat maps or box plot that you saw. That's where it comes from. And of course, there are many other associated resources to many experiments, such as bedcraft files, RDR files I talked at the beginning, images, also raw files in TSV form that you can download if you want to perform your own analysis. Now, these 3 sources are split in terms of data size. So in blue, we have the Postgres database. In green, it's the file storage, and in red, it's the Solr indexes. So this is the original files, the original data size that we were talking about is about 600 gigabytes for the last release, and that's post-processed and filtered to provide this, which is exactly what the application uses to run. Now, in terms of our technology stack, for the backend, we use Java 8 application on our Tomcat server. We use also Spring and Maven to control the build life cycle of the application. And for the front end, we use React, Webpack Babel, and NPM. So the usual pattern that we have is a React component that consumes a JSON payload that's produced by an endpoint from the backend. So we can say that in terms of backend, we're a bit more conservative, but in the terms of in front end, we are a bit more daring to work maybe with experimental tools. And finally, the pipeline that we use to process the experiments when they come from ArrayExpress and deploy on Atlas is a mixture of different scripts orchestrated by Bash, but we use different technologies such as Scala, R, or Perl. Mostly, we try to use the right tool for the right job. We're not very dogmatic in that sense. So some of the challenges that we face when developing Atlas, one is to keep up with the ECMAScript or JavaScript development ecosystem. So every week there's not just a new version, but a new tool that has taken the whole web by storm, and then you have to or check that, or maybe left with a deprecated application. Solar itself is, for us, it's a huge challenge. As Luca has shown, it offers a great deal of customization, plugin development options, and we really learn here as we go. Also have a lean build workflow, which is related to the last point, really, which is modular deployments. So right now, we try to have a release every month and a half, 2 months. Unfortunately, when we have the data, the code is quite coupled to the data, and that is a challenge, because then one of the things that we need to work on is to actually be able to release data without releasing code, and vice versa. And last, I would like to, of course, say that this would be possible without our team, Robert and Irene as manager and product owner. We have the curation and training and outreach group. Laura here is here. She presented also a poster on Expression Atlas. Unfortunately, Amy and Maria are no longer with us. Again, as Damien said, they're not there. They're doing just fine, but they moved along in their careers. We have Nuno and Sohib are two bioinformaticians, and Haider, Wojciech, Elizabeth, and yours truly, parts of the software development team. So, without further ado, thank you very much for your attention, and I'm be happy to take any questions that you may have.
Thank you, Alfonso. We can take one question. Somebody got a question they particularly want to? Okay.
Right.
I think everything was very, very clear there. So thank you, Alfonso. That's really great. Okay.